Hello students, welcome to my channel. In this class, what we do study about modification protection footprinting. Before this, we learn or we study about the nucleus protection footprinting. So this is also the protection one. That is modification in what? Nucleus part. So how? We'll see. Chemical cleavage agent can also be used instead of nuclease. That is the modification. We use here not a nuclease but some kind of a chemical which work as a DNase I. So, modification protection footprinting is similar to DNase I. So it is similar to DNase I footprinting, but difference is we are using here chemical. So the basis of the technique is the only protein binding of DNA part of nucleotide sequence is protected from the modification. So that part which is protected from or protected with the help of protein of a DNA sequence which is protected from a protein by cleavage in the nucleus. But here it is protected from a modification because we do here modification. So where protein is bounded on the DNA, this part will save or protected from the modification rather than nucleus cutting. So instead of DNase I digestion, the DNA fragment are treated with limited amount of base modification agent, which is very important, dimethyl sulfate. Remember that dimethyl sulfate is the chemical which is used instead of DNase I, and instead of cutting DNA it will do what? Modification of the DNA sequence where protein is absent or protection by the protein is absent. And where protection is present or protein is present on that particular DNA sequence, that part will escape from the modification. So DMS, dimethyl sulfate, adds methyl group to the G nucleotide. This is a DNA sequence or strand where so many sequences are present. But this modification only done on the position of where the G nucleotide is present. If G here, G here, G here, G here, then the methyl group will be added here. So it is a DMS. If a protein bind at particular DNA sequence, if this part G is also present, but this is protected from some kind of protein which is protecting from the modification and cleavage both type of things. So here there is a G nucleotide, but still it cannot get methylate or methyl group cannot attach to that part. The guanine at that site are protected from the DMS action. In the DMS modification protection assay, fragment are treated with लिमिटेड डीएमएस, जैसे कि वहां पे हम लिमिटेड ट्रीटमेंट दे रहे थे डीएनएस I का, as similar we are treating limited amount of DNase here for modification. So the only one G nucleotide at one fragment is methylated per fragment. So there is per fragment is cut at once a place. So here also one segment only modify at a one G nucleotide. It is similarity but different. How it is different? You know it. A radioactive end label is required for visualization of final product on the gel electrophoresis. As we see that nucleotide or DNA strand was used as radioactive. Why? So we can visualize easily what is happening here. So here also we do radio labeling at the end. Why? We'll see. One portion of DNA is combined with protein and another portion are kept free of the protein. We take a DNA strand where is no protein. As we see in last one also, we make the 2 copy of this DNA segment and then 1 copy will be free from the protein and 1 copy will mix with the protein. And then the other step of the experiment will be done with same condition will apply on both A and B line. Okay, then both sample are treated with DNase I with modification of nucleotide G. Now we use to mix DMS here and DMS here also. So we can see where the G protein free. Here all are free, no protein modification. Here some places where protein attachment or bounding specific place is there. So only protein free area will be nucleotide modification by the methyl group. DNA region bound with protein remain protected from the DMS as protected from the DNase I, and this is how it is protected from the DMS modification. That is, and there is DNase cleavage. After removal of protein, DNA is treated with piperidine. So after removal of protein, no. After doing this treatment, we will treat that DNA with the piperidine, which cleave phosphodiesterase bond at modifying nucleotide position. So this only cut from where? Where the methyl group is present. Only cut that part of the DNA segment where the protein modification is occur. Where the protein is present, that is protected from the methylation. So it is also protected from the piperidine cleavage. And what is piperidine do? It do phosphodiesterase bond cleavage. It will cut phosphodiesterase bond. And piperidine cleave only modified strand, not produce double strand. It will cut only if there is a G nucleotide and this is modified with the M, and there is a G protein, so G nucleotide, and this is also modified with the methyl. So it will make cut from this part only. So this part will cleave this region only, and here this region only. This is not cutting through the double strand, only cutting single strand. DNA fragments are resolved by denatured gel electrophoresis. And then the denaturing gel electrophoresis, what it will do? It will cut from this and separate this segment and this segment and this segment and this segment in the gel electrophoresis and position it according to their sizes and separate two strands. So there is no need to separate two strands with the help of any enzyme. So it can be denatured or separated by the gel. And label fragment of free protein bound DNA. One is free and second one is protein bound DNA and is labeled. Are compared on denaturing sequence gel visualized by autoradiography. So and are labeled in both on two strand, one gel electrophoresis and second. One have protein free and one have protein bound DNA segment, and that is labeled with the radioactive elements. So we can visualize the what we have to visualize. We only visualize where the strand is present. So we can compare these strands with these strands. So we can find out this is the particular region of the sequence of the DNA where protein is binding. So resulting autoradiography. Why autoradiography? Because we are using radioactive labeling, and in radioactive labeling there is always or most of the time we analyze or visualize with the help of autoradiography. This is how autoradiography will be done is also I explained already, and there is a video on the YouTube channels so you can see this also. Shows stand with one labeled and one and generated by piperidine cleavage. One is labeled in initial and one is cut with the piperidine cleavage. Because why? One is labeled and because there is a protein, so there is no cutting with the help of piperidine, but another is showing the cut fragment or a small fragment. So we can be autoradiograph. This is not important line one. The bending pattern of the control DNA, which is without the protein, indicate the position of G nucleotide in the DNA fragment and footprinting. So without protein is the controlled one. Remember that. Not the mixed protein or protein present or protein bound one is not called a controlled one. Controlled one is without protein, because we are doing what? Studying the presence or specific site where the protein is binding. So protein binding is compared with the free of the protein sequence. That's why free one is called a controlled one. So this is indicating the exact position where the G protein is bound with the G nucleotide is the bound with the specific protein, and that fragment will called footprint of that particular protein, presence of protein. Now it is a interference assay, and that is also a modification interference. We'll see in next class. Okay, bye bye.